Hi, my name is Eri. Today I'm talking about the skeletal stem cell anatomical heterogeneity in the craniofacial bone. In 2015, we discovered the presence of mouse skeletal stem cell, which possess the ability to self-renew and differentiate the bone cartilage and stroma. While we have previously characterized in femur, we have not investigated to characterize the SSCs in craniofacial bone. Therefore, this study, the purpose is to identify those characteristics in the craniofacial bone. In this study, we segmented each skull from frontal, parietal, occipital, nasal, and mandibular bone, and then digested with the buffer and then FOX isolate the skeletal stem cell. In the panel C, I first conducted the coronary formation assay and then identified that in the D panel, the coronary formation ability was highest in nasal-derived SSC, followed by parietal, mandibular bone, and occipital bone, and frontal bone. The frontal-derived SSC has a very little coronary, so we excluded the frontal bone for the further study. In a panel D, you can find that the osteogenic capacity, which is the top row of the panel E, was really high in occipital-derived SSC. While the nasal-derived SSC have a high chondrogenic activity, you can see in middle part. And kidney transplantation assay further validated this functional capacity difference. For example, you can see the blue staining in the nasal-derived skeletal stem cell, which is a cartilage formation. And then under the occipital bone, you find a bone formation, which is orange staining. And in FGH, the characterization, the quantification result of the E. We further investigate the transcriptomic variety of skeletal stem cell from each part of the bone and then described in figure I. The figure I, for example, is osteogenic related gene. But you can see for example, the mandibular-derived SSCs has a high SP7, which is the osteogenic driver gene. And this is corresponding very well with the functional assay. In the J, you can see the chondrogenic gene modulation. And you can see the SOX9, SOX6, which is chondrogenic drivers genes are highly expressed in the nasal-derived SSCs shown here. And to conclude that, we identified the functional variety in skeletal stem cell in craniofacial bone, and then this is also coming from the transcriptomic level. In the future, we will validate this finding using the in vivo Caraballo transplantation assay. Thank you very much for listening.